First, I just want to thank BBRF for the opportunity to come talk as a young investigator. We young investigators are always very enthusiastic to get a chance to share our early results at this early stage in our career. My research focuses on looking at the brain at the molecular level. I really am hoping that by looking at the molecular level, that will point us to the mechanisms that underlie schizophrenia. And today I'm going to be talking a little bit about our research in looking at whether inflammation plays a role in schizophrenia. We have reason to believe that inflammation or change in the way that immune cells are functioning does play a role in schizophrenia, but unfortunately, even advanced methods in detecting markers of inflammation in tissue from patients with schizophrenia have really yielded inconclusive results to date. And the discrepancy among the results may be related to the tissue types that are being probed. So markers in blood might not necessarily reflect what's going on in brain. One of the other issues that often comes up is that studies may differ in the clinical population, so that some patients might be on medications in some studies that are not being used in the patients who are studied in other studies. And the medications might be having an effect on the immune response. So we aim to address the enigma by using complementary techniques, biochemical testing of the most relevant tissues, cerebrospinal fluid that bathes the brain, and looking at the brain itself using molecular imaging techniques. We'll start with this model. So we think that there might be a role of oxidative stress, that's overproduction of free radicals that might impart damage on the brain, as well as an abnormal immune response, together causing altered neural connections and circuits at a particular point in neurodevelopment, and that this alteration in neural connections ultimately causes the cognitive, behavioral, psychotic symptoms characteristic of schizophrenia. And this model is based on work by many who have gone before me, but also based on results of an early study which I led a team who looked at CSF, cerebrospinal fluid, of patients in their first years of schizophrenia. And we found that there was a great decrease in a particular antioxidant that points to potentially there being oxidative stress playing a role. And then we found an increase in interleukin-6. Interleukin-6 is a signaling molecule that regulates the immune response. And so again, that pointed us toward this model, and we've been pursuing it by looking at an even bigger sample of CSF that we've collected. We have a sample of 35 CSF specimens from healthy controls, 46 patients with schizophrenia, and it's important to note that the patients have all never been exposed to antipsychotic medications. And then we also have CSF samples from 15 individuals who are deemed to be at risk based on either having a family history of psychotic illness or having what's deemed to be mild symptoms that don't meet full criteria for schizophrenia. And when we look at the CSF of the patients, oh, hold on, jumping the gun. There we go. Let's see. There we go. Again, interleukin-6 is increased in the CSF of the patients. When we look at the at-risk individuals and compare the results to that of the controls, there is not. In fact, there's a decrease in the level of interleukin-6 compared with the healthy controls. So it might be something that's specific to schizophrenia as a disease. Interestingly, when we look at the receptor, interleukin-6 receptor is decreased both in people with schizophrenia and in those at risk. And the other thing is that when we look at other inflammatory cytokines, this is the only one that seems to be increased, and other pro-inflammatory cytokines either were not able to be detected by the assay, which I think is a major problem, or were unchanged. And so we're continuing to look at this CSF sample population using more sensitive assays to see whether we can actually see other pro-inflammatory cytokines has changed along with the interleukin-6, and then also looking at markers of oxidative stress. We hope that these results will better help us to understand results that we're also getting from probing the brain directly. I'm probing the brain directly by using molecular imaging. So what is molecular imaging? It's the visualization, characterization, and measurement of biological processes at the molecular and cellular level in humans. It's imaging biochemistry in real time in the human brain, which is why we're so excited that we have this technology. So just so that we're all on the same page, we heard Wayne Drevitz talk a little bit about PET scan results. We're using positron emission tomography, and we're taking a compound, that's way over there on the left, that binds specifically to our target molecule. In this case, I'm going to be talking about a protein that's changed. It's increased in its expression when the brain is undergoing injury or repair. That compound binds to that protein, and because it's radio-labeled, when it's injected into the patient, it goes to the brain, binds to the protein of interest. That radio label emits a signal that the PET scan can pick up, and then we can visualize in 3D where that protein of interest is located. This is the protein of interest. It's called the translocator protein. Translocator protein is a mitochondrial membrane protein. It's actually one of several proteins that sit on the outer mitochondrial membrane, and it's of interest because in states of microglial activation, so microglia are the resident immune cells in your brain. When they become activated because there's brain injury or repair, they greatly increase the expression, the amount of TSPO that's sitting on their mitochondrial membranes. And so the technique in using PET imaging that detects this amount of protein is based on the fact that if there's more TSPO, because there's more microglial activation, we're going to see more signal on the PET scan. That's actually not what we see in schizophrenia, to just cut to the chase. So we're looking at the particular region called the hippocampus. We think it's very important in schizophrenia, and so that's why all the white arrows are pointing you to the hippocampus. This is a healthy control participant, 2 different visualizations of his PET scan results. And this is an age-matched patient with recent onset schizophrenia. Again, 2 different views of his PET scan results. And it's subtle, but the bright red signal here in the hippocampus of the healthy control participant is not there in the patient with schizophrenia. And we can go a step further, because PET scans are quantitative. We can take data from 11 patients with recent onset schizophrenia who've undergone these PET scans. And I'm going to walk you through this because it's a little complicated. It's color-coded because the patients and controls in blue share the same genetics for the translocator protein. So I'm going to have you just look at the blue individuals, the blue data points. The open circles are our healthy controls, and right here, the crosses in blue are the patients with schizophrenia. And what we're looking at is the binding, designated as VT, the amount of binding in the hippocampus. And the patients have less binding in the hippocampus than the healthy controls. And then we add in the green individuals, not because they're green, but that's how I've colored their data. They, again, share genetics for this translocator protein. And again, the patients with schizophrenia have less binding than those of the healthy controls. And we can look at statistically all of this data together, controlling for the effective genotype, and we see that there is a significant difference, a significant decrease in TSPO in the patients with schizophrenia. So what does that mean, right? I was expecting when I walked into this to see increased TSPO, increased microglial activation. Again, 2 points. Translocator protein is a mitochondrial protein. And if there is oxidative stress occurring in schizophrenia, it might be damaging the mitochondria and causing a change, a decreased amount of translocator protein to be expressed. The second point is that translocator protein, by being decreased, may just be pointing us to the fact that the immune cells aren't functioning correctly or might not have the apparatus to function correctly. So in summary, the two results that I've discussed, we found a change in the concentration of markers related to inflammation and oxidative stress, namely interleukin-6 and its receptor, even in the CSF of even medication-naive patients. And we're now testing for other markers of inflammation and oxidative stress in that CSF sample. And we see decreased binding of this radio tracer that binds to translocator protein in a critical brain region, the hippocampus, in medicated patients with recent onset schizophrenia. So the future is that we, because of NARSAD funding, we're continuing these studies to look at additional CSF markers of both inflammation and oxidative stress to better understand our ongoing PET image data of TSPO, and also to inform future PET imaging data of other markers of the immune response. I want to acknowledge and thank our research participants. And there are many, this is interdepartmental work going on at Hopkins as well as with collaborators in Germany. So many people to thank. But I just want to underscore one more time how important it's been for me this year to be the recipient, I guess in 2013, of the NARSAD Young Investigator Award. It has been responsible for allowing me to get all of this pilot data, from which we're applying for more funding, and it's also generated a lot of enthusiasm amongst people who work with me, and now I have young colleagues wanting to come join the team, so thank you very much.